Ministry of Defense. So I'll start by a brief overview of my career path, which universities I've been working in, before introducing you to the work I've done before arriving in French Polynesia. And I'll finish by presenting the work I'm conducting here and the future projects I'm hoping to develop here. So it all started with a master project on the foraging behavior of elephant seals of the Kerguelen Islands before my PhD that I did at the Museum of Natural History in Paris, where I studied the evolution of dispersal-related behavior in butterflies. But today I'm going to tell you about what I did since then, so since my PhD, which started with that postdoc at McGill University in Canada, where I worked on the evolution of cognitive behavior in birds from the Caribbean. I then moved to Sydney, Australia, where I had the chance to work on cane toads, so a species that's really a model species in evolution, and I had the chance to study population both in the invasive range and in their native range. I then moved to Barcelona to work in Daniel Sal's lab. I've been collaborating with Danny for a few years already, and it was a good opportunity to finalize some projects we've been conducting together before finally spending a bit less than a year in Daniel's lab at the University of Cambridge in the middle of COVID. And I finally joined the UMR here in French Polynesia as I obtained my position at the IRD almost 4 years ago now. And so this pathway means that I had the really great opportunity to work in universities from Europe, North America, in Australia as well. I also had a chance to conduct fieldwork in a diversity of environment, including in many islands from the Caribbean to Hawaii, and obviously here in French Polynesia. And this path has given me the chance to value my work through different ways, both by publishing my work in a series of scientific publications, publishing book chapters, communicating about my work in international conferences, but also through invited seminars. Also to act as an expert by reviewing articles, reviewing projects, both research and conservation projects, but also putting a lot of effort into trying to transfer this knowledge, but also talking about the current biodiversity crisis with the public in general, either via interaction with the media locally, nationally, internationally, but also with a lot of interaction with NGOs and directly with the public via public conferences. So I worked by giving conferences to NGOs, but also by developing scientific support to their project, especially here in French Polynesia. This has also been, since I arrived here in French Polynesia, an opportunity to involve myself in the development of a collaborative project. We have here a research unit that's very diverse, that's working on subjects that are very different, very transversal. So it's a great opportunity to build this transdisciplinary project. And I've tried to push for the development of these projects with colleagues here in the research unit. And all these career paths have also been an opportunity to participate in the supervision of students, especially undergrad students so far. And I've co-authored 15 articles with either PhD or master students so far, as I believe a demonstration of my involvement in the supervision of these students. And since arriving here in French Polynesia, we've been very lucky to profit from the funding of a lot of different agencies, which enable us to develop a lot of, I believe, really interesting work. And I hope I'll convince you of this at the end of this presentation. So here I just listed the funding agency that helped us since we got here 4 years ago. So with that, I'll jump into the science of this work I've been conducting. And very originally, for an evolutionary biologist, I'm going to start by talking about this guy, Charles Darwin. So Charles Darwin did not invent the theory of evolution. Evolution was already an idea that was there, had been developed by other people. But what he brought in was, among other things, one key mechanism that really solved some big issues in that theory. That's the mechanism of natural selection. So natural selection was kind of solving a key question, which was how do we explain the match between form and function in organisms? This observation that organisms are adapted to the environment. That was an issue at the time. We didn't have a good mechanism to explain it. Natural selection explained that pretty well. And that's a mechanism that's been largely integrated in a more integrative theory of evolution that was developed in the 20th century by people who work on what we now call the modern synthesis of evolution, that gathered all these new results, especially related to molecular biology, to genetics, into a more integrative theory. But natural selection remains a key factor in this theory. And that theory is still the basis that we use today as evolutionary biologists to work around and understand the evolutionary processes in the world. It is a relatively easy and simple theory to understand, and that's probably one of the key reasons why we use it as a reference. It considered that there are four major evolutionary processes: so mutation, drift, migration, and natural selection. And I'm going to mostly focus on natural selection today, because that's been really something that shaped the work and research I've been conducting over the last 15 years or so. So the conditions for a trait to be under natural selection are also pretty straightforward. You need the trait to be variable. If there's no variation, there cannot be selection. All the individuals are the same in the population. There's no one is going to be selected rather than another one. That variation needs to be heritable. If you don't transfer this variation to the next generation, it also will potentially just disappear, not be selected. Finally, that variation needs to provide some kind of advantage in terms of the production of offspring. So you need to have an adaptive value to this variation for it to be selected. And you need these three conditions to be fulfilled for natural selection to occur. And this sounds relatively straightforward and relatively simple, making of natural selection what might emerge as an easy mechanism to study. However, the more we work on these concepts, the more we work around this, the more we realize that everything is more complicated than it sounds. We know that variation is more complicated than it actually looks. The selection is not just acting on one trait, it's acting on the structure of this variation and its full complexity, on association of traits that might be associated because of adaptive processes, because of constraint acting on them, etc. Heritability is also more complicated than it sounds. This theory, the modern synthesis, is mostly a genetic theory of evolution, but we realize today that there's more than genetic that is heritable. There is a lot of non-genetic mechanism that can be transferred from one generation to another. And there's also non-heritable variation that might be important and shape some of the evolutionary processes. We're going to talk more about that in a few minutes. Finally, one challenge and an opportunity at the same time for us evolutionary biologists is the fact that the conditions today are changing drastically. We're right in the middle of a massive biodiversity crisis. We're going through potentially a new mass extinction crisis, meaning potentially more than half of the species that are on Earth today will go extinct unless we're changing things. And this is all related to the activity, the human activities. But the point is that it means that the environmental conditions, the selective pressure that are acting on organisms on Earth are changing drastically. So if we're trying to understand how current organisms have evolved, it is a bit tricky because we're not in the same conditions as the one in which they evolved. But also is a great opportunity because it means that we can look at how evolution is happening live in response to these changes. So it's around these more complex mechanisms, more complex patterns that I've organized most of my research over the last 15 years. So the first axis I've worked on, I organized this talk following the organization of the manuscript. The first axis I worked on is the structure of this variation. What is basically the material on which selection can act? And I mostly focus on this idea that traits are not evolving independently from each other and try to determine whether there are associations between these traits. I've done that on a set of different organisms, but mostly on birds. And I'm going to show you one example of the work I've conducted on these questions, which I've been working on the cognitive traits in birds, try to understand whether different cognitive performances were associated within these birds. We just realized that if you just capture a few birds from the same species, the same population, they will potentially be very different in their performances at different tests. And this variation is maintained in nature, which raises the question, why is selection not selecting some that are potentially more performant, or on the contrary, less performant if it's costly? We realize that this variation is existing, and one reason might be that there are just different strategies that are equally valuable, equally adapted in these ecosystems, in the ecosystem where these populations are stranded. So to test this question, I just kind of scratch the surface of it by exploring the association between a set of different cognitive traits to understand if there is some structure in these traits. What we did is just measure different cognitive performances in these birds. So here we just brought birds in captivity for a few days, and we looked at how good they were, how quickly they were able to figure out solution to different tests that we are offering. So here on the left, you have a bird that needs to figure out how to open a box to access food. On the right here, they need to be able to associate a color with the presence of a reward. So two very different cognitive mechanisms. And we also tested a bunch of other potential traits in these in captivity. And what we realized that these traits are not just varying randomly. You don't either have some individuals that are extremely intelligent and others that are pretty bad at everything. You have some compromise, some trade-off between these traits. Some are pretty good here. You can see on this figure on the left, some are really good at solving this problem you were seeing on the left earlier when they were opening the box. These ones are pretty bad at learning to associate a color with every word. And it seems like these different cognitive performances are associated along a speed accuracy trade-off. So there's actual cognitive strategies that are coherent that we can detect here. And not only are they associating different cognitive traits, they're also related to other traits, including physiological traits that you can see here, for an example, on the right. What we're really showing with the results is that if you want to understand how natural selection can act on variation, you probably need to understand how the traits are associated together. The selection is probably acting on these suites of traits rather than on individual traits. So in parallel with this research, I've conducted a lot of work aiming at better elucidating the importance of non-genetic mechanism, non-genetic origin of variation. So I work on parental effects, so the fact that the parents may transfer resources, transfer information to their offspring that will have consequences independently of the genetic on the phenotype that these offspring will develop. I worked a little bit as well, thanks to great collaborators, but I'm not a molecular biologist, on differences in gene expression. In that case, it's not the gene sequence, it's not the actual sequence of DNA that matters, but which genes are expressed and how that will shape some phenotypic differences. And that's also a key parameter that will drive a lot of variation, even though it's not directly genetic. And finally, and that's what I'm going to go in more detail about today here, I worked a lot on the importance of developmental plasticity, both in shaping evolutionary processes, but also as a source of variation to adapt to your environment. So, phenotypic plasticity is the ability that individual genotypes have to produce different phenotypes when they're exposed to different environmental conditions. Just to give you one example, you see these two tadpoles here. There's one that was raised in presence of predators, and they developed this large tail that allows them to escape predators more easily. In contrast, you have this other tadpole, similar genotype, was not exposed to predators, it did not develop that tail. And the reason it did not develop it is because it doesn't need it to escape predators, and if you develop that larger tail, it induces a cost. Later in life, they will have a reduced size, slower development, and a higher probability of not surviving through that tadpole stage. So you don't want to express that response to predators if there's no predator around. It's better to have a plastic response where you just express this phenotype only if the predator is around. And interestingly, this phenomenon of plasticity seems to be pretty ubiquitous in organisms in general. And today, the theory is suggesting that potentially it also plays an important role in bringing new genotypes and favoring the emergence of new traits eventually through the evolutionary process. So the idea behind that is that a change in selective pressure, just like the changes in the environmental conditions we're experiencing today, may trigger a stronger selection that will potentially eliminate most individuals, but some may already have a solution via a plastic response that will emerge faster than a fixed response via mutation. So it's faster to have potentially that plastic response already present than to wait for the mutation to emerge in your population. Importantly, though, these plastic responses we've seen with that example can also be very costly. So eventually, across time, that plastic response may be assimilated, and will slowly erode towards a more optimal genotype that is not that becomes canalized. We talk about canalization here. So under this theory, hope I'm clear enough with that. Always a complex process to explain. Feel free to ask more questions about this aspect later. But under the theory, we expect both plasticity to help individuals survive under environmental changes. If you're more plastic, you may have a better chance to develop the phenotype that will be the right one quickly. But it may also allow to usually occur later on. So it's kind of a buffer for evolution to happen before you go extinct when the changes are occurring. The issue is that this process is really difficult to characterize because it's very ephemeral. Most of what we're looking at is a result of this. So we're just looking at different genotypes here. It's really difficult to understand whether they result from an initial genetic mutation or from something that went through a plastic response. And this is where cane toads are really interesting. So cane toad is an amphibian that originates from South America and has been from there introduced to many islands mostly, and was introduced to Australia in 1935. And in Australia, really interestingly, since its introduction, it hasn't stopped colonizing new area. It is still colonizing habitat that are potentially suitable to it, but Australia is so vast that it hasn't reached the end of it pretty much. So it means that there are cane toads today on the invasion front here in the west of Australia that are into a 90 years of invasion, so a numerous number of, 90 years of colonization, so lots of generation of colonizers, so also very interesting evolutionary processes that are associated with that invasion process. So originally these toads were introduced as a biocontrol for these beetles. You can see on the left they are eating the cane, the sugar cane, with very little effect on these beetles, as very often in these stories, but a lot and massive effects on the native fauna of Australia. The reason for that is that all the life stages of these species that goes from the eggs through the tadpoles to adult toads are poisonous, and the predators in Australia have not evolved with this type of toxin, so they're very sensitive to it. And I just want to attract your attention here on one short stage, this hatchling stage during the life during the life stages of the toad. That's what's going to be particularly interesting for the next slide, which is that moment where the larvae is emerging out of the eggs, it's hatching out of the egg, so it's not protected by the egg anymore, but it's not able to swim yet, so it's not able to escape from predators. So it's a short period that lasts 1 to 2 days, and that's where these hatchlings are very strongly exposed to potential predators. So that period is going to be really important in a minute, you'll see. So what we realized is that when these tadpoles and toads were brought to Australia, these came with a big change in selective pressure. They're in a different environment that I was mentioning. The predators in Australia are not really adapted to control these species, because they die if they eat toads or tadpoles. That means that the population of tadpoles are much higher in density here in Australia than they are in the native range. So there's a change in that selective pressure, more competition between tadpoles. There's more tadpoles per pond here than there were in the native range. That has triggered, we believe that was a trigger for the evolution of a cannibalistic behavior that was already existing in the native range. There's some cannibalism. Larger tadpoles will eat these hatchlings, so at these stages where they're really sensitive, but to a lesser extent than they do it in the introduced range. Interestingly, in the introduced range, not only are they cannibalizing more these hatchlings, they also are actively attracted to them. So if a female is laying eggs in a pond that already has tadpoles, it is really likely that at the moment these eggs are going to hatch, they're going to be eaten by the tadpole that are here. It's not the case in the native range. So this evolution of a cannibalistic response is that massive change in selective pressure on the development of these tadpoles that has triggered the evolution of an interesting classic response. So I'm going to take you step by step through that process. We first looked at how tadpoles in French Guiana, so in the native range of the toad, are responding to the presence of a cannibal. In that case, what we did, we just put these eggs and these hatchlings in boxes that have or don't have the smell of an older tadpole, so a potential cannibal, and we look at how they react. Do they accelerate their development, which might be a good way to decrease that sensitive period and escape the potential cannibals as fast as possible, or on the contrary, do they not do that? What we found is that in French Guiana, in the native range, where the selective pressure by cannibals is very weak, so there's much less cannibalism, there's all kinds of strategies that are co-occurring. Although the dominant one is just an absence of response. If you put the tadpoles, the hatchlings in presence of the smell of a cannibal, they just don't do anything. They don't change their development, which is probably a good strategy because they are really unlikely to be eaten anyway. But there are some that are plastic, either in a way that we could call adaptive, they accelerate the development, that this red line that you can see at the bottom of this figure, and these ones accelerate the development, probably are more likely to escape the cannibals, but this comes with a high cost later in life, because these guys that accelerate the development will eventually transform into much smaller tadpoles in a few days later. So there's a cost to this plastic response that they will pay later, which the hatchling that are not responding to the cannibal are not paying. And you got another strategy that's much rarer, where a few clutches were, on the contrary, slowing down their development, which is probably not a very good strategy to respond to cannibal. But interestingly, these responses have changed in the introduced range. So if we're looking here at the middle panel, that's what's going on the invasive front in Australia, where we have much stronger selection from cannibals. You're much more likely to have cannibals in your pond that will actively try to eat you if you're being laid in one of these ponds that are invading from Australia. And what we found is that there, the only strategy that seems to prevail and exist is that strategy where the hatchlings are actively responding to the smell of a potential cannibal. So no matter what, they will always respond to the cannibal. It doesn't matter how costly it is later in life. The reason for that is probably that it's better to pay these costs later than just to be almost guaranteed that the entire clutch is going to go eaten. And where the story becomes really interesting from an evolutionary point of view is, or even more, should I say, is that in long-established population in Australia, so towards Queensland, where we've had continued selection within these new environments for generations, and the selective pressure by cannibal is really high, we realize that not only these plastic response still exists, so the tadpole, the hatchlings, it's a tadpole for hatchling, the hatchlings are going to accelerate their development in presence of cannibals. But there's a new strategy that emerged as well. And it is this cannibalized strategy where by default the hatchlings are developing very fast. It doesn't matter if they're exposed or not to a potential cannibal, they will develop very fast. And this is probably ultimately the best strategy for them here. We just needed plasticity to give the time for these to emerge so that they can respond to these cannibals. So these strategies are probably ideal. You avoid the cost of the plastic response, but you're developing really fast, no matter what. In any case, you're really likely to have cannibals in your pond, so it's probably the best strategy to have it. This was a key example that showed that phenotypic plasticity can facilitate the emergence of a new genotype in this system. This is the rapid development of these hatchlings. So we provided with that story a unique evidence, I believe, of how phenotypic plasticity can be a driver of evolution in the wild. What we are bringing is more question, though, about the importance of plasticity in these evolutionary processes at a global scale, how important has it been in history, but also on the potential role that plasticity may play today for species to respond to environmental changes. And so the next two axes of my work have focused on exactly this type of question. How do populations, how do species respond to the changes that they're experiencing in their environment today? So I did that at two different scales. I worked at a population scale where I mostly compared populations of birds and amphibians in habitats where they are exposed to different pressures, especially different environmental pressures related to human activities, mostly urbanization. And what we found here in most of these work was that behavior, mobility, cognition, condition, all these traits vary substantially across habitats. So these animals from the same species are very different when you're measuring them in these different habitats. They seem to respond strongly to this exposure to these different conditions, these strong environmental pressures related to human activities. I'm not going to go into detail with these studies here. I'm going to pass to talk more about the next scale I worked on, which is at the scale of the species. So with a similar question, but instead of comparing population, comparing species. Why are some species doing better today than others when facing the massive changes that we are experiencing today at a global scale and a local scale? So to that aim, I've used a lot the IUCN Red List, using these indices of extinction risk that determine how likely a species is to go extinct in the near future, giving them a score of probability of extinction, and try to determine whether we can predict this probability of extinction with a set of species characteristics that would potentially allow us to better understand which species are sensitive to these changes and why they are sensitive to these, really elucidate some of the mechanisms behind. So to that aim, I've run comparative analysis on large numbers of species. So the idea is to really determine if we have global patterns here. That allowed me to confirm at a broad scale that niche breadth is a really important factor. So species that occur in a broader diversity of habitats are doing better than the ones that are highly specialized, which makes sense. If your habitat is destroyed, but you can move to another one, you're probably going to do better than if you cannot. Life history also was shown here as being an important factor, especially some particular life history strategies like root parasitism seems to provide some advantages to species here. Intraspecific variation, that's also something that's expected in theory. If you already have a lot of variation within your species, you probably have more solution to cope with the changes in your environment. So we could confirm that using polymorphism as a proxy of variation, color polymorphism, so the fact that within species you can have diverse color morphs that coexist, and that's also a proxy for variation within the species. And finally, and that's what I'm going to tell you more about today, I work on the role of behavioral plasticity. That's something that's kind of in the air, has been in the air for a few decades, but that's never been formally tested at a global scale for multiple reasons. So why would we expect behavioral plasticity? So this ability to change your behavior in response to a variable environment or to changes in your environment. Why should it be helpful? Well, the key response here, to my opinion, that the first thing that an animal can do in front of a change in its environment is alter its behavior. So if you're able to alter your behavior in an adaptive way, you're probably better off than if you have a very stereotypical behavior, no matter what your environment is like. So we imagine, especially, that this could be helpful for species exposed to these different conditions, these new conditions, because they may be better able to take profit from new opportunities, new food items that emerge as a result of these changes. And they may be better able to switch to new resources if their previous resources not available anymore. And they may also be better at adapting their behavior to the arrival of new species in their environment. Avoid, for example, being active when new predators form. However, we also find a lot of potential counterexample when we're thinking about these highly behaviorally plastic species. Some of the species that come in mind are lots of primates, lots of parrots that all tend to be doing very poorly today and be at very high risk of extinction. So does it really help to be more plastic in your behavior? Does it really provide an advantage? And so we were hoping to test this at a global scale to answer that question without having to focus on these potentially anecdotal results that suggest that it may not be. So the key issue here was to measure behavioral plasticity at a broad scale so that we can compare that, give a score of plasticity to different species and to species that potentially are very different in many aspects. Well, this is where it was very interesting to work with Louis Lefebvre and with Daniel Stoll, as that's exactly something they've been working on before. So what Louis had realized was that birders and ornithologists from across the world are really keen on describing interesting behavior they're observing that birds are expressing. So some unusual things that birds are doing in the wild that are not in their usual repertoire. And this, Louis had that key idea, realizing that these are pretty much plastic responses, new behaviors that emerge in populations, innovations. That's what we're using in that case. And by just going through the literature and collecting all this information on unusual behavior that had been observed and reported by the scientists in the literature, Louis could build that huge database listing all these examples of innovation. And that gave an opportunity to give a score for each species of how many times it had been reported being innovative in the wild. So it was an operational index from wild population, not measured in captivity. This is an index that's really focused on foraging-related behaviors. So because that's probably the most easy to observe, and that's where most the higher diversity of innovation have been reported. Of course, we know that this parameter is not without biases, without issues. First of all, the species that are more often investigated are more likely to be seen being innovated. If you have more researchers focusing on a given species, they're more likely to report these information. So we try to develop different ways to take into account these potential biases, including here by developing an index of research effort to correct so that species that are more investigated are expected to also have more innovation than the ones that are less investigated. We also ran a set of studies to try to understand exactly how innovation, innovativeness in general, can be integrated into ecological strategies, into life history strategies, and are part of these general suite of traits that species are expressing. And some of these studies, we were able to test exactly what we wanted, which is to say, is behavioral plasticity affecting the risk of extinction as a proxy for the response, the species response to current environmental changes. So we built this type of model where we're trying to look at the impact of innovation on extinction risk, population trend, using these broad indices from the IUCN, and trying to understand whether innovation matters once we've taken into account a set of potential bias and parameters that could confound our efforts. And we found with this study that indeed innovativeness is one of the key characteristics that predict the risk of extinction. Species that are more innovative have a lower extinction risk and are also more likely to be on the increase. So we added behavioral plasticity to the set of tools that species can use to respond to changes in their environment. And with this, I'm going to move to the work that we've been conducting here in French Polynesia with this last axis that's really inspired by this general diagram you can see here and aims at working on one specific environmental change that is having a massive impact here on islands, which is the introduction of invasive species. We've been developing different projects aiming at better understanding both the eco-evolutionary dynamics around these species, and also how to improve the status of these ecosystems by managing these species. I want to underline here that all the work I'm going to talk about now is work that we've been developing together with Jane and the board. And so I would not have done half of what I'm going to present here without Jane being here. So it's really a shared, fully collaborative work that we've been. Just to give you a bit of context, the reason why we jumped into these projects here. So after working on these global indices of the IUCN global extinction risk, we could have these interesting results, interesting patterns, understand potentially new mechanisms, or at least give some cue about them. But we also had these very depressing results every year that you just add more and more species to these red lists. The risk of extinction are increasing for all these species, and there's not much you can do about it. You're just looking at it and you're just a witness of it as a scientist. I realized that with some threat, there are things we can do about it, and we were really hoping to develop a more applied research in addition to the fundamental approach that we developed. And it turned out that islands and invasive species were a key area to develop these kind of groups. Islands represent a very minimal proportion of the Earth's landmass, but an excruciating large proportion of the modern extinction. Here we're talking about extinction in terrestrial vertebrates. And they also are hosting most or a very high proportion of the currently the species that are currently at a very high risk of extinction. And most of these both extinct and at risk of extinction species are impacted by invasive species. Sometimes only invasive species. Invasive species are sufficient to explain their extinction very often. And here in French Polynesia, we realize that not only invasive species are a big problem, but we also are in a hotspot of extinction. So globally, we estimate, at least via the IUCN, that we lost about 164 bird species over the last 500 years. So species that are extinct. Among these 164 species, there is 14, so more than 8% that went extinct here in French Polynesia. They were endemic from French Polynesia. So that's a huge proportion, given that French Polynesia represents about 0.001% of the surface of the emerged land. And today we still have a very high proportion of species that are at high risk of extinction. So it turned out to be a very hotspot of extinction, and most of these extinctions are due to these invasive species. Finally, we also realized that there was very little local research in the terrestrial environment here. So that's really something that was missing, and potentially something where we could add, bring something relevant to try to improve this situation. So with that in mind, we developed a set of projects with this general theoretical diagram, aiming at understanding what's going on with invasive species specifically, addressing both fundamental and applied questions. With invasive species being at the center, what's going on when we add invasive species or when we remove them? How do populations, communities, ecosystems, meta-ecosystems respond to that? Looking also from the population to the land sea connectivity as well. And all of this work we are conducting it in this eco-evolutionary dynamic framework. So this idea that ecological patterns are affecting ecological processes, are affecting evolutionary processes as well, and conversely. We also aim at taking context dependence into account. So the results we have here, we can compare them across context and can potentially extrapolate them to other areas than just our site of study. And of course, the purpose of this is also to develop more applied research, help prioritizing conservation action, and develop control and eradication methodologies for this invasive species. So we developed these ideas into two main frameworks, Petiaroa and the terrestrial bird communities of French Polynesia. So let's jump into Petiaroa. So Petiaroa is this atoll about 50 kilometers north of Tahiti, right in the middle of the Pacific Ocean. And it's an atoll that's particularly interesting because we have 12 different islets. So it was many replicates to run an experiment in a way. Now of these 12 islets, there's only one, the one that's at the bottom left here, that's inhabited. The others are conservation land in a way. Very interestingly, as most islands in French Polynesia, lots of invasive species have been introduced here, but we have 3 islets, the one that are here on the right, that were never invaded by rats. So we have 3 control islets with no rats, and all others were invaded by either just Polynesian rats, so small species of rats, or both Polynesian and black rats. And when we arrived here in 2021, the Petiaroa Society, so the organization that's running the restoration of this atoll, so the whole restoration program around this atoll, was already starting a rat eradication program. They removed rats from 3 islets, and they were about to remove them from other islets. So we had the time to collect 1 year of data before the eradication on these 6 extra islets, and we're still today monitoring what's going on since then. How is the ecosystem responding to this? I'm not going to go into detail with that, but we've also been working on the eradication of another problematic species, so the yellow crazy ants that you can see in yellow here was also used to be, because we've almost finished eradicating it on 4 different motu, and we address here very similar question, because they all had a similar impact to the rat impacts. And so this figure is pretty much summarizing what we are hoping to document. So can we, from the situation we have on the left, get to the situation we have on the right once we've removed the rats? Why do we expect that? Basically because we're working on these coral atolls. So we're on a coral cemetery. There's just dead coral everywhere. And there's very little nutrient, so very low productivity potentially, except when you have seabirds playing there that are bringing their nutrient via the guano, bringing nitrogen, phosphorus, etc. And they favor a more dynamic environment. They favor the growth of the vegetation, and they also bring the nutrient that will flow into the surrounding lagoon and affect as well the health of the marine ecosystem. So far, this is a process that has been mostly developed, and a theory that's been mostly developed using correlative studies by comparing islets with islets without rats. Here we have a chance, because the rats are being actively removed today. We could collect data before the removal of the rats to look at what's going on after the removal. So the Synthèse Society has brought together a pretty amazing consortium of researchers from all over the world to investigate that, to understand how the atoll is responding to the removal of that 1 species or these 2 species of rats, by bringing people that are working from the microbiome of the coral up to the vegetation on this motu. What we're bringing in here is precise data on how the seabirds are responding to these changes, mostly, which I hope that I convince you that seabirds are expected to be pretty much a mechanism by which everything is going to change. At least that's what we're hoping. We're also looking a lot at what the crabs are doing, the terrestrial crabs, because we know that these species are also at the center of a lot of the ecosystem functions in these atolls. There's no mammals that arrive here naturally, no native mammals. So most of the functions that are usually being taken care of by mammals are here being taken care of by these crabs that are also very sensitive to the rats and other invasive species. So in this framework, we've developed a research project that had a series of different objectives, one of them being to quantify this variation in the guano input from the seabirds, clearly one of the key aspects of that work. Another is to really set up that long-term seabird monitoring program, which is a unique program in the region. We're in the 4th year. We're really hoping to make it sustainable in the long run and keep monitoring these birds. We also, as I was mentioning, looking at what crabs are doing, how they're responding to the removal of rats at different scale, from the internal variation to the community changes, and also at the role they're playing in the functioning of this ecosystem. Because even though we know they're important, we don't have much information on how important they are. So this has been the opportunity to train and supervise different students addressing each specific question related to these different objectives. So that's also been a key aspect of this research. And so in parallel, the objective was to, via these studies on the crabs and on the bird, as it still is, to understand how the ecosystem functions are changing, how the functioning of these ecosystems are changing. So that's one aspect that we're working on, while also aiming at understanding how the changes in seabirds affect the lagoon by collaborating with marine biologists. And finally, just going to mention that here we're also working on this ant eradication, where we're both developing the methodology that we are importing from Australia, actually, while testing its secondary impact here on the non-target fauna. And we're really happy that both seems to be working very well in Aotearoa, and already the methodology we've set up here is being used in other places of French communication. And again, this has been the opportunity to train students, and very recently we were very happy to have been able to get Michael to stay with us to conduct his PhD, specifically to address questions related to the functioning of this ecosystem and how these ecosystem functions are impacted by invasive species. That's since Michael arrived in October. And just to finish with Aotearoa, this is also a pretty amazing place to work, because the Synthèse Society is working with that triptych of how research is informing restoration programs, and in turn education as well. So we're working with schools that are coming to Tetiaroa, schools from Tahiti, from Moorea, with students as well that are coming from Tahiti, from the US as well. That's a great opportunity to basically present the work that's being done there and show that you can do something about the crisis we're going through today. I'll jump into the last part of this presentation, which is our second main project that we're developing here, and that focuses on terrestrial bird communities of French Polynesia. So as an ornithologist, I imagine French Polynesia as basically being some kind of paradise when you're going to see amazing birds everywhere that are unique to French Polynesia that you can't find anywhere else. There are all these endemic kind of legendary species that are here in the area. Yet when you're in Tahiti, you don't see any of these guys. If you want to find them, you need to go deep in the valleys and be lucky enough to have a chance to find one that's still present on the island you are. In contrast, you see a ton of birds, and all these birds have been brought from pretty much anywhere in the world, except Northern America for some reason. But so these are just the species you can see here, are all the ones that were introduced with success and that still are thriving today, but lots of other species have been introduced and eventually went extinct. So this is both very sad, but it's also a really interesting opportunity for an evolutionary biologist. Because we have these introduced birds that have been put together almost like an experiment finally. And not only have they been introduced to Tahiti, but they've also been moved to other islands, or sometimes they move naturally and colonize other islands naturally. That gives us an opportunity to look at, for example, what's going on when you have an island with only the common myna, or what's going on when you have both the common myna and the bulbul, or none of these two species. So you can look at different combination of species, understand how they interact, how they are affecting their own ecological and evolutionary processes, and this ends up being a real pretty amazing experimental setup in a way. So we really aimed here at developing research in that general framework, but before jumping into that, we realized that we needed a lot of basic information on these birds. But unfortunately, very little is known about them. There's been a lot of work to know which island has what species, so really inventories to understand where these species are, but that's about all there is. There's also a lot of work on some species that are at a very high risk of extinction by NGOs, but that's really the extent of it, especially from the science part here. But we realized that the first step was to develop this information, bring this information so that we know how many individuals there are, what are the relative abundance, what are their habitat preferences, what kind of, when are they breeding, what kind of food are they collecting, etc. And that's true for both the native species and the invasive species. If we want to better understand how these invasive species are impacting the native ones or how they're evolving here, we need to know well both their ecology and their natural history. We also aim at bringing information on their very basic natural history here. So how did we do that? We've developed a whole set of protocols, but mostly we've been working using these point counts, where we're just counting birds in different areas across islands. So we've done over 1,200 point counts on 18 different islands. And the idea that with these point counts, we have good estimations of relative abundances of these different species, and allows us to know which habitat they favor. And we also at the same time have information on their basic behavior. We're collecting more information than just the number of birds, but also we're looking at what they're doing, what substrate they're using, etc. So this is really bringing a lot of very basic information on what these birds are doing and how they are impacted by different gradients of disturbance as well. At the same time, we're developing some methodological questions. We're addressing some methodological questions because these protocols to survey birds are mostly being developed in areas that are very different from the habitats we're in, from these islands here. And also because we often have facing some challenges, some constraints that we don't see in other places. We can be just dropped on an island for a few hours. We don't have any control on when, for how long we'll be there. And we want to know if, when we're doing a point count between noon and 1 p.m., what can we get out of it? Is it even worth it? And if it is, how should we rate that to interpret it and compare it to this point count that we do in more classic moments where we imagine that the birds are active. So this has also been the opportunity to collect a set of very basic information on the birds that are poorly known just because there's very few people studying them here. That can be just information on the spread of introduced species that we observe on islands where they were not known to be, but also records of new species that were not recorded in the region. And for example, last year we had Emma Kilbu, who developed a whole project aiming at better understanding how the common myna is using different habitats in Tahiti by comparing their behavior across habitats. But so ultimately, the goal of this work is not just to better understand how these birds, what these bird ecology is, but also to both address evolutionary question and bring some solution for conservation purposes. So the first aspect that we would like to explore, and this is really something that's beginning thanks to arrival of Antoine in the lab a few months ago, not even 2 months ago actually, as a PhD student. And the purpose of it here is to really use that natural experiment, well, not quite natural actually, but that experiment in situ to try to test a set of hypotheses with regards to how bird song is evolving, how the acoustic behavior of birds are evolving. We can, basically the environment has been manipulated for us. These birds have been put on some island, not others, and we can look at how they change their calling behavior, their acoustic behavior in general. That's one thing that we just started addressing with Antoine's PhD. And in parallel, we're hoping to use these acoustic tool that are being developed. That's one of the reasons we're focusing on the acoustic, that it's a new strategy that we can use, a new methodology we can use to monitor birds, including in areas that are difficult to access here in French Polynesia. So we know that these passive acoustic recorders are not like perfect, but they provide a great advantage here, where we have 124 islands, some very remote that are difficult to access, and potentially we just can set up another recorder there, come back a few months or a few years later to extract the data. And we can do that again a few years later and collect data that are directly comparable, directly standardized. So that's one issue we've been facing here in French Polynesia as well, is that not only there's little studies, but when several studies have been conducted on the same islands, usually they use different protocols, so they're difficult to compare. It's hard to determine whether the populations are actually changing or it's just a protocol issue. So with this tool, we're hoping to bring something new that could favor a more accurate study of how these birds are doing today. So we're hoping to develop, the dream would be to develop this acoustic observatory based on the example of the Australian acoustic observatory. So for that, at the moment, we've just started developing collaboration with different NGO and some towns as well, the Tuamotu, to hopefully, in the long run, start something more sustainable in the long run, where we'll really settle these recorders into. Now, I'll finish with this last slide, which is not really a conclusion slide, but more of an introduction to how we want to develop these research programs in the future. And our research philosophy as well, and student training philosophy to some extent. So I'm really happy to talk about that during the questions after this. So one first aspect we want to keep digging further in is the use of invasive species to investigate eco-evolutionary processes. They're really a unique model to understand evolution, especially contemporary evolution. And so we want to keep working on that. We would like to add one new aspect, which is the study of native population of species that have been introduced here in French Polynesia. We've seen that with Kenko, it has been very rewarding to have the chance to look at what it looked like in the native branch. I think we bring really interesting elements. Also want to keep developing these partnerships with NGOs and local townships, etc., to favor the restoration of habitat and improve the resilience of local ecosystems and local communities to the global changes. Also clearly want to make our monitoring program long-term. So especially I'm thinking about, of course, these acoustic observatory I've been mentioning, but mostly as well the seabird monitoring that we're talking about in the Chihuahua. And finally, that's something we've been developing more and more recently, putting more emphasis in the collaborative work and more integrated project where we are taking profit of this very strong dynamic today on the territory in French Polynesia between research groups to develop integrated research on the land-sea continuum in areas that matter for Polynesians, or in areas where most Polynesians are living. Think about Tahiti, which is a pretty understudied island. And all of these projects, we really aim at developing them with in mind that our job here is not just to provide data, but it's to build a local capacity to help French Polynesia reach its objective in terms of mitigating the impact of climate change, decreasing its emission, preserving its ecosystems, but also help them making informed decisions. We're not there to make decisions for them, we're there to help Polynesians make the decision that are most suitable for them. With that, I will finish by thanking, first of all, again, my committee members for accepting to take the time to review this work, but also to all the people that have contributed to this work. I didn't list them because I was clearly going to wait. I'm going to forget probably dozens of them. So I'd like to thank all the collaborators, all the students, all the volunteers, the colleagues, the administrative staff, of course, and the technicians and funding agencies. Without any of these people, none of this could have happened. Thank you so much again for listening to me.
The reviewer questions. Okay, so maybe we could get, so if we could stop the sharing and we can see the other members. Please. Perfect.
Change the camera.
Yeah, it's gonna be. And then if we have that, it's tall enough to reach them. There.
Sorry.
That's okay. I think affiche. If you go to that affiche thing, maybe up at the top. You know, that's like so much information. No, like in a good way. So we're gonna start with questions by the reviewers, and reviewers, you have about 20 minutes to ask questions and have a conversation. And we're gonna start with Claire. We're just going in alphabetical order. There is no order of importance. It's just alphabetical. So Claire, go for it.
Okay, thank you. Can you listen? Can you hear me well?
Yeah, perfect.
Oh, okay. So thank you very much for inviting me. I didn't know your work so before, so it was really a real pleasure to discover all what you did over these 12, 15 years of research. And I was really impressed by the diversity of approach and questions that you took, and you seem to really quite passionate with you were working for a few years or something and going deep and then changing. So maybe it was also because of postdoc and things like that, but it was super nice. I'm also very impressed by what you want to do now with this island, and I think it will really give some very important, basic, fundamental, and evolutionary answer. So and I think you have a really great design, and it's super nice to have all these islands that you work with a big array of people, and so I really look forward to see what you will find in the upcoming year. And so I'm going to ask a few questions. First about cognition. So I'm not. So I wrote most of my questions, so it's more a matter of discussion in my report. But I have one on cognition because I find cognition really something intriguing and really nice to work with. But I'm always puzzled by the fact that it is for us as humans very difficult to ask the good question to the animal, and if we don't ask the good question, then it's quite risky to relate it to fitness or to do comparative analysis. And so I'm sure it's something that you thought deeply about it. But I wanted to ask you, what are your fear and what are the things you really trust, and what will be the advice that you will give to people that will start cognition on new species?
Yeah, that's a really interesting question. I don't have a clear answer to that. I definitely agree that, I mean, it's just like in psychology, the way you're asking the question is going to influence the answer that you get, and it works when you're testing a cognitive threat with a bird. Just one story I like to tell about this is how we were trying to measure the string pulling performance of bird, which is a classic test where the bird is sitting on its perch, and there's a reward at the end of a string. and it basically needs to pull the string and pull it with its beak, and then pull it in its legs to pull it again until it can get to the food. So it requires a lot of coordination and then relatively good understanding of how to do it as well. So it's pretty complex. But testing that on these grackles, you could see on videos earlier, was pretty tricky because they were never doing it the way we wanted them to do. They always find another way to respond to it. So they're not actually doing the behavior you want them to do. So we had to change it multiple times. Some would just, flutter next to the reward and access it without having to do that complex thing. Others would just shake it as much as they could until eventually it broke and they could get it at the bottom of the cage. So we ended up having to design different devices here to really make sure that it could only respond to that test the way we wanted them to respond. But it had an impact in that, indeed, in nature, they don't really face that. They can always try to design different solutions around it, and that's probably one of them, but it doesn't mean that it has to be the one they're using. So I 100% agree. It really varies depending on how you ask the question. I don't have a strong advice, except that I believe you should always take the point of view of your bird, because his purpose is not to do it right, it's to get the food, basically. So that's what he's going to go for. And it's going to be affected by lots of other aspects around there, including, how hungry it is, what is his general boldness, what is his fear of new object, or how used to new object he is. That will affect as well what you're testing in captivity, but also in the wild. So I think that we don't have a perfect way of measuring it, but we have ways to try to take these potential biases into account and try to improve your way of testing bird. I think each species has to be tested in its own way to some extent. You need to know your species very well to be able to design the proper test here. I don't know if I'm responding well to that question. But
yeah, no, it's a really an open question. I of course don't have any answer to that. It's really something that I always wonder when I see cognitive tests, and because I think it's super nice and important to get access to this data. But and as you say, maybe when we know well a species, maybe we manage to ask clever question. But when we want to do comparative tests, it's another layer that is even more tricky, because as you show with innovation, so also it's even more complicated to compare the tests. So also I think it's really valuable, but I don't know how to do to be caution and how to discuss this point in papers, pushing people to, I don't know, to know better the hearing capacity, visual capacity, or to know better the species in the wild, to ask also, to ask them to do something that they face in the wild, to ask also a test that is meaningful for them. I don't know. So it was also this kind of tricks that maybe can be discussed, or I don't know. I don't know, when you go in meeting where people, like a symposium or yes, workshop where people are working on cognition, what is discussed in this meeting in general? Yeah.
One aspect that's being discussed a lot is that this area of research is pretty young, actually, because it's recent that we connected that to behavioral ecology. It used to be more psychologists that were running these kind of tests, and so we've had the point of view of psychologists a lot in this area of research, and that's changing now, because we're looking at these performances not as much as what is it able to do or not, but more as how adaptive can it be and how does it relate to what they're actually doing in the wild. But still, when you're measuring their performances during these tests that you're doing in captivity or in the wild, by default you're changing what they would do naturally, because you want to test what they're doing. So it's always a challenge, I think, between measuring some characteristics that are pretty abstract, including for us, but also for even more so for the bird, but also try to relate that to active behavior, actual behavior that they have in the wild, and connect that to more realistic trait.
And doing more of these cognitive tests in the wild, do you think it's a good thing, or it would be too variable, and because we want to put the bird outside from other environmental factors to detect something? Or do you think there are some people that are doing tests in the wild, opening, I don't know, trying to enter somewhere when the entrance is blocked or something like that? I don't know. What do you think about it?
I think it's super complementary. You can address very different questions as well. Indeed, like here in captivity, we had this advantage that we could control the level of hunger of our individuals. We would just stop feeding them for a given number of hours, and we knew that each individual was in about the same condition, had been spending the same period in captivity before, which is something you don't control in the wild. But at the same time, what you're looking at in the wild is more realistic of what is happening there. So one aspect that people have done a lot is pretty much blocking the entrance to a nest box for, I'm thinking about great tits or blue tits, for example, and looking at how good they are at figuring out a way to open that door. And this, I think, is really interesting as well, brings different information. It's probably way more dependent on the motivation of the bird as well, but there are biases in both cases. I think they're addressing slightly different questions, and both things are probably complementary. I feel like what we're doing in captivity is probably bringing more fundamental question on the structure of these cognitive traits rather than more behavioral ecology question that can address in the wild more easily.
What do you mean by structure when you say structure of the trait?
Like the trait we've measured in these grackles are very artificial in a way. Like it's not clear that our grackles in the wild need to associate that yellow box with a green box very often. But if you just block their nest box, it's probably the case that sometimes there's something that will hide the entrance of a nest and they have to find a way to get in there. So it's probably a more realistic competence that you're measuring there.
And I was wondering, you work with this cognitive test as you integrate behavioral syndrome and cognition, and you seems to find cognitive trait to be part of behavioral syndrome. I was wondering whether you often found that in most species, or whether it was quite rare, or how from all the studies that you made at the end, are you convinced that it is a part of the behavioral syndrome? And also, I was wondering about sample size that you need to be sure about that.
Yeah, no, that's a great question. I again don't have a clear answer to any of these two questions. I haven't worked on enough species to be confident enough to say there is a general pattern, and we can tell that behavior and cognition are always strongly associated, and these coherent strategies are universal. I don't know that we have this answer yet. We need more data on other species, probably. Remind me, what was the other point that you were asking?
I was in most of your graph. Your sample size were not super high, and I was wondering whether you will recommend to work with bigger sample size, or whether, because there are more and more papers that find that when you have a slow, this meta-analysis that comes out now very frequently, and that shows that it's difficult to replicate some of the studies that we are doing, and then I include myself in behavioral ecology, and especially because sometimes we go for too small sample size and we find result by chance. And I was wondering, what is your feeling in the field of cognition for that? Do you think people are well aware of that, or we still need to be a bit more cautious?
Yeah, I think we need to improve that a lot. And I think one of the key reasons for that is, well, I don't want to criticize the psychology colleagues, but the origin of this discipline was psychology. And originally, a lot of the work was to demonstrate that the bird was capable of doing something. So it was sufficient to just have one individual doing it to demonstrate the species had that capacity. And here we are kind of inheriting that to address more broad-scale questions, I think, that are not really centered on that are more evolutionary and focus on the evolution of their behavior. In that case, we need indeed larger sample sizes. One challenge is that these cognitive tests take a long time to run. Here in the work I've conducted, we went for the option of only working, mostly working, at least on these grackles. It wasn't true on another species, on these bullfinches. But on grackles, we decided to just capture our birds all in the same area to basically look at the structure of these variation in these cognitive traits within the one same population. So more than the sample size, I think that we need replicates to test whether, say, a population that we sample in a different habitats or on a different island would show the same structure or not. Here we just wanted to look at variation within the population. So that was, I think, sufficient. But for a more advanced research, we would need indeed much larger sample size.
Yeah, thanks. And then I had a question about, so you managed to, it was super nice to see how you managed to correct for research effort but at the end I was wondering whether it is possible in because we know nothing about most species of the world in tropics and in many places. So I don't remember if you restrict your if you think that if you run your analysis I don't know, only in North America and in Europe, it would be the same result, or do you have a bias that is triggered by the fact that even if you correct for research effort, you cannot correct for everything, basically?
Yeah, no, it's a very good point. Indeed, research effort is just one way to take these biases into account, but we're not removing them completely. What we've done is that we ran a separate analysis after excluding species that had a research effort that was really low, to make sure that we had species that were sufficiently studied so that we could expect for them to have a recorded innovation if they were innovative. So I don't remember exactly where we put the threshold, but I think we decided that if the species had more than 15 papers on the Web of Science database, it was considered as being studied enough. So that's one way we dealt with that. Initially, we ran the analysis specifically on, as you mentioned, North America and Europe. The main issue we had, especially for European species, is that are not so many species that are at risk of extinction, at least at a global scale. So we had a low variability at that with that regard. So I don't remember exactly what the pattern was, but that was an issue. The other thing that we did is that we included the geographic area as a random effect in our models to try to take this into account in addition to the research effort as well. So, you know, our random effect includes North America as a different category than Europe or Africa or Southeast Asia, etc. So hopefully we're taking that into account to some extent.
Thanks. And I have a last question about cognition. I saw that you use brain size sometime as a proxy of cognition, and this is really debated also. And also, I have the same question about after some year of study, would you recommend? Because it's a very handy trait when we want to do comparative study. But do you think would you recommend? Does it bring something, or do you think we really need to be cautious about when we use brain size?
I think we should use it, definitely. I think it's a great trait. I think it's been tested and validated multiple times in birds as a very relevant in this index. You need to correct it by body size because there's a strong allometric relationship. Larger species have a larger brain, but once you've removed that effect, brain size is a strong indicator of different indices of cognitive capacity. We know that the global brain size, so the size of the entire brain, is tightly connected to the size of the specific part of the brain that's responsible for these more complex cognitive processes. So it's indicative to use a whole size of the brain. There are some exceptions. There's some species, I'm thinking about brood parasites or species that cage food that have some part of the brain that are overdeveloped in an area or another for these specialized behaviors specifically. But on a global scale, it's a really good index. It's been criticized a lot 15 to 20 years ago. And I think that these critics are pretty much removed today by new results that have been developed, especially by Daniel Saul's lab and Will Lefebvre's lab, that have also demonstrated not only that the size of the brain, like species that have a relatively larger brain, also have a more dense network of neurons. So there's more neurons per cubic centimeter in a bird that has a larger brain. So it's clearly an indicator that there's been selection on their brain capacities, not just the size that they increase, it's also the content. And we also have recent work by Jean-Nicolas Audet, who's one of my collaborators in the US, who's shown that the ability to solve problems in captive birds, I think he had 15 or 20 different passerine birds, several hundreds of individuals that he tested in captivity, and could show that the variation between species in their ability to solve these problems was tightly connected to the size of the brain as well, and to innovation. So it looks like this is all part of the same story. The main reason we didn't include brain size in that large-scale study on extinction risk is because we know that it's strongly associated to life history, and we wanted to avoid being biased with that regard and just have a result that would just mean it's not actually cognition, it's a life history that's explaining our results. So the correlation was too strong there, so we tried to avoid that in that case.
Okay, thank you. So I think I have no more time for questions, but I will ask you just two small ones. So, and one is more about your HDR. It's so what do you want to share with students and to give to them? So what do you expect and what will be your challenge, you think, with them, and your big joy, and so what is your position? And
Yeah, thank you for that question. What I'm really hoping to share is my passion. I would say our passion, because we were really supervising the students with Jaina together, and we really are trying to share the passion we have for the work we're doing, but also our concern for the state of current biodiversity, especially. So that's what we're really hoping to share, more so than the ability to do science in itself. The science is also really important, and that's what we're pushing for, of course, as well. The way we're trying to train these students is really to adapt to each student. Each student is totally different. Here we have students at the University of French Polynesia. We also are training students that are coming from France. We're training students from the US as well. They all are from different backgrounds, different histories, and we're really trying to adapt our way of training them to the students. One aspect that I really liked is how Louis Lefebvre, the professor I worked with in Canada, who was just telling me how he was training students. He basically has some students that arrive there at this level, and his objective is to bring them here at the end of their internship. But some of them arrive here, and the purpose is to bring them there. You don't aim at for all students to arrive here. You just aim them at making progress. You want them to get something out of their internship and get better at doing science, doing research, and at caring for the environment. I think that's how I would respond to that.
okay, thank you very much.
Thank you. Move on next to Patrick, please.
Thank you. Thank you so much. Thank you, Simone, for inviting me to be on this panel. And thanks everyone here. It's great to meet all of you remotely. So Simone, I really appreciated the opportunity to get to know your research better in reading your manuscript, I guess it's called, and then a lot of the related research that you've done. And honestly, I'm just really, as I mentioned in the summary, I'm just really impressed at the quality and the volume of the work that you've done in a lot of different areas, all related to phenotypic plasticity, of course, and most recently on its effect on animals' ability to adapt and how we can better conserve animal populations. I'm impressed with your research output, your scholarly contributions, the journals that you've published in. It's all really impressive for, the stage of your career. Honestly, you're just getting started, it seems like, by a lot of measures. And so I'm really looking forward to potentially collaborating with you on some of these, some of your research goals, particularly as it relates to bioacoustics and how song varies across islands and how it's going to change, and how we can use the song, how we can use birds' behavior, and particularly to monitor their population size and changes in density and so forth. So I don't really have any particular research questions to ask you. I think what I really wanted to know is just a little bit more, and these are just the question that Claire asked towards the end, but just a little more focused on your mentoring philosophy for students. If you could just maybe summarize the students that you have mentored in the past, and you gave a quick summary on your presentation, the types of students, and then your sort of plans for students in the future. And if you could just kind of let us know about a little more detail about your philosophy for mentoring students and working with them to help them achieve, their goals, which is what a lot of what we're doing here is trying to train the next generation of biologists and conservation scientists. So I'll stop my question there. If you could just talk more about your mentoring history, philosophy, and then plans for future students.
Yeah, sure. Thank you, Patrick. And I'm also looking forward to developing collaboration with your lab. That would be a great opportunity for us. Yeah, I'd gladly talk more about our mentoring philosophy with students. So we've mostly supervised students actively since I arrived here in French Polynesia. So since we got here in 2021, during the first year, we had one student, Milena Philippe, who's been working, so was a student from the university here. He's been working with us in Tetiaroa. So that was the first experience for us. I'd supervised master students, but during my PhD, so 15 years earlier, it was coming back to the French system as well. So understanding how it works. And these master programs are very short in France. So it's a 6 months project, meaning when the student arrives, you need to have a project that's ready to go because they need to design an experimental protocol, run the fieldwork or the experiments, write a paper and run the analysis and present the results within even 5 months here. Actually, it's really very fast. So the way we're trying to do that, since that first experience with Milena, that was a great experience. And we then had 4 students the next year. We had 3 last year. We have 2 students this year, in addition to the 2 PhD students that just started. What we're doing is that we really aim at kind of making them believe that they are the ones developing these protocols because they arrive and we want them to go through that exercise of writing the protocol and developing it then themselves. But we know pretty much what they're going to do because these programs are too short. It's very different from my point of view to then train a PhD student because in that case, they have more time to do it. And we really want them to come up with their own protocol, with their own questions as well. So that would be, I think, the big difference I see between these two types of students. The other aspect that we are trying to really put forward is that we are trying to be very plastic in our supervision of these students by adapting to the student, not just to their level, but to their personality, to their interest, to their way of working. So there are some students with which we're going to give them more time to read, to try to assimilate things from the literature before making decisions with regard to what they're going to do. Maybe we already know what they're going to do next, but we'll still give them this time because we believe that it's beneficial for them. With others, we feel like they're just ready to jump in right away, and that's what we do with them. We try to really adapt according to the person here. There are some students that we are interacting with almost every single day, if not more than every single day. That's happened over the last few years. There's others, 1 meeting per week is sufficient, and they are really much more independent. So we're really trying to be very flexible with that regard. So far, at least, maybe a bit less flexible in the future as the projects are accumulating. But so far, that's how we really prioritize the work that the students are doing over our own projects that are independent of us.
Thank you. I think, yeah, I think I have sort of a similar philosophy in working with students, but yeah, thanks for that. I really don't have anything else, any other research related questions for now. So.
That's okay. I think it's okay if we move on, if you're okay with it. Yeah.
Yep.
Okay, excellent. Okay, we'll move on then to Pierre.
Yeah, thank you. So thank you, Simon, for inviting me to be on the board for your habilitation to supervise research. So we were at the same line when I was recently recruited and you were doing your PhD. We knew at that time we had something in common, but we didn't work together, and then we split, we didn't meet again. And it was such a pleasure the day you wrote me to ask me if I could be reviewer for your habilitation. And when I read it, so impressive all what you did. All the approaches, the levels, the methods you did. I think about the 4 postdocs you did, I think I applied to 2 labs I would have dreamed to go, and you did it, I didn't. So really, really, I was very happy to discover all what you did, the complementarity, and we have so much in common, so thank you. Thank you for this time. And the time I took and discovering what you did and your thoughts. Can you tell me around what percentage of your papers you cited in your manuscript? Are they all in there, or there were still some that you did not really include it? Just the overall, not the exact proportion.
There are some I didn't have space to cite in there, and that are really side project that did not integrate very well to my general story that I put in there.
Okay, no, it's for the public, because when you read this manuscript, like in one page, he's summarizing like 2 papers, and there are 112 pages, and it's so dense. So the first pages, you read them twice, and then you say, I never finish. So if I read each page twice to assimilate everything, so it's so rich and very interesting. Um, um, I was also impressed with what you developed at the end of your presentation. You put the focus on what you are doing now, these projects, and there, it, again, it's for the public and the order of the jury, is to realize that you move to new field. Now you're in functional ecology. You are going to fluxes, litter decomposition. You are also launching acoustic monitoring. You never did nothing of that, but no problem, you go. Just you have like this confidence to say, yeah, let's go to new field. I learn and we'll develop that and we'll cover the whole Europe with because you showed us that it's the scale you will be working at with bioacoustic recorders all over Europe on your initiative, coordinating the others. Wow, it's just it's impressive, and when we read it on the paper, we believe it will work. Given all what you did, we trust in you. So yeah, that was I appreciated this this ambition, and the others already have outlined the relevance of what you do, the understanding adaptation to environmental change, but also with very fundamental questions on evolution and ecology. Um, I don't know where are your limits. So then I jumped to one question about, again, the philosophy for the training of the students. Because if they have a look at your CV, it's like the Himalaya. They have to go to climb if they want to impress you somewhere. And so how do you guide them to go where is their will, their future, because it seems that you can offer them nearly everything. So how do you take someone, chat with him, and then say, okay, we'll go there with you?
Yeah, I mean, I almost want to say, come back in years and I'll tell you more about that. With master's students, as I was mentioning, we basically have a written project before they start. We know already what they're going to do. So in that case, there's not much flexibility in the actual material that they're going to create, except the quantity of it. With the recent experience with our two, so with Antoine and Michael, our two PhD students, we wrote the projects together for both of them. With Michael, the challenge was that he too was interested in everything, so we had to figure out what we should do or not from these projects. And so we had to be selective on the few species he would focus on. And I think I would be amazed if he had the time to do everything we put in that project. So I think the way we're doing it at this stage is just to keep the opportunities open and see within the first few months of the PhDs where he's going to go and what he's going to focus on. He's already collecting a lot of data in different framework that we identified together as being really interesting and very promising. So that's the way we've been working so far. But that's potentially some aspect that he won't have the time to treat. And with the other student, with Antoine, so far he's just been there for months, and so far each meeting there's more and more added on his plate potentially. So I think that's the next step here is for him and for us to help him focusing on where he's going to go specifically. But I think I really enjoyed myself during my PhD having these few months of just reading papers, understanding what the literature is about, what's been done before, to develop my own project. And I base a little bit that philosophy today with these students on my own experience. And of course, if I realize that really in another month or two, Antoine is still considering broadening again his project, maybe to include other things than bird or stuff, we'll put a stop there and focus the project a bit more, but we're not there yet.
Okay, so now I'll go through some specific questions that I picked while reading your manuscript, mainly for curiosity, but also to exchange ideas on. So the first one is very broad, and it's a thought I had one day, and what I wanted to share it with you, and to see how you react, or maybe you thought about that. Also, is that one day I was wondering. I said, and I'm interested in phenotypic plasticity, but then you have the phenotypic variation, and I was thinking in general the partitioning of this phenotypic variation into what is plasticity and what is genetic driven. Right? That's a very basic question, and then it came to my mind just to think that the higher the cognitive abilities, the lower the room for the hard drive, for the genetic drive, because then the higher you have flexibility, your behavioral flexibility through your cognition abilities, the more you can compensate for the limitation of your genes. And so then you have very basic organisms. Well, I mean, small, very hard drive organisms, and the more you have cognitions, then the more you have variation, phenotypic variation, so including behavioral variation, that is soft driven, and by what you learn, condition during your development, and and then you were, with your results, you showed us that behavioral plasticity is one element that reduces the risk of extinction. And then I thought, so that is indeed behavioral generalism, and it's again the same story that generalists are winning in this changing world. So have you thought about that, broad, about the link between hard-driven and soft-driven phenotypes and how they partition throughout the live beings, and this link to the capacity to adjust either by evolution or compensation adjustment?
Yeah, definitely. That's something we've talked a lot about when writing that behavioral plasticity paper and when working on this evolution of a plastic response in these cane toads as well. This is a very conceptual conversation. We're probably going to lose people there. But clearly, one aspect that I find really interesting, and some of the work you've done, I think, was also emphasizing this possibility that plasticity is kind of a buffer to respond to changes. And that's one idea that we've been developing, especially in the toad paper, that it pretty much gives you more time to adapt in a more hard way, as you're saying, via genetic mutation. The key question here to me is whether it is a general thing or not. And I think that we're not really able to capture that at this stage. Our measures of behavioral plasticity, I believe, are not strong enough to be able to disentangle between these mechanisms. I don't think that we're really measuring generalism in that case, because we're even how much of that is behavioral plasticity could be questioned to some extent. We're measuring the ability of these species to show new behavior that haven't been observed before. So let's say that these are actual innovations that are something new that emerged in these populations. Is it something that emerged in response to a change? We don't really know. We're not taking that into account in our database. We're also only focusing on the foraging innovation. There's plenty of other behavior that matters. Just the one that for which we can't have an operational metric here because of the sufficient amount of data. So I'm not sure that I'm responding very well to your question, but I think that we're kind of capturing something different than the generalism, also because we've tested in a couple of papers, we looked at how innovation is associated to these ecological generalism, and it is associated to some extent to both diet generalism and habitat generalism, depending on which metric we're using. But it's still a different thing. There are associations, but it doesn't mean that they're totally independent, that they're totally dependent either. So yeah, I don't have a final answer to that, of course, but that's an interesting conversation. Yeah.
Okay, thank you. Now I'll go to more technical questions. One was in the in your interesting studies where you want to disentangle the effect of the research effort, well, to adjust for the research effort. And I was wondering how you accounted in that for the relationship between phylogeny and distribution range or areas on Earth. More specifically, I have the feeling that a large part of the specific radiations are in the tropics and then colonized more towards the pole for several groups at least. Of course, there are others that have their diversification core outside from the tropics, but still there is a collinearity between diversification and the area you are, and the research effort is not the same in this area. And so, yeah, I wanted to know if you tried, how do you feel you achieved to get rid of this collinearity between phylogeny and biogeographic ranges?
I think we did to some extent. We did to some extent by including indeed the phylogeny in our analysis, by also including biogeographic range. We did not fully exhausted the issue there, of course. One thing that we did, though, I don't think that we included in the paper at the end, is that we also ran these analyses separately for the different parts of the range. I think I was mentioning that in response to one of the questions by Claire, just to test for result consistency.
Yes.
We did not necessarily distinguish that in our actual analysis. Well, the only thing I can tell you is that one advantage we had is that we were working at a very broad scale. I think we have over 8,000 species in this database that we were analyzing, and that allowed us to have basically, even if some of these parameters are associated, the associations are very weak. So we have collinearity, but with factors of collinearity that are pretty negligible. So just a few %. So based on that, I'm relatively confident that this is not impacting our results, but indeed there's always space for improvement in these analyses.
Yeah, okay. Also, I really like the paper or set of paper about habitat breadth, and I felt it's promising and original. It was innovating to try to develop this index. And you decided to build it on the IUCN habitat definitions. I understand that then there is this direct link, so you have some kind of homogeneity because then you work with the IUCN status for the trends and some other stats that they aggregate. But why are there other reasons that, other than the homogeneity of the database, to build an index for habitat breadth on the IUCN habitat definitions? Because I felt that there are more richer, more objective ones that are developed elsewhere, because IUCN primarily developed variables for the purpose of expertise of the conservation status, not for scientific purpose.
No, you're definitely right. I definitely agree. This is not necessarily the best solution. The reason we use that is indeed so that we could work at such a broad scale. So in that analysis on habitat generalism, when we developed that index in that specific paper that was about the index, I think we have over 20,000 species of vertebrates. And I don't believe that we have another index that we could use for that other information on the use of habitat than that one. We will reuse that index in a paper with to remember the name of this group, and in a French team with Céline Teplitsky and I forgot the first name of the students, Martinossi, where we use this exact similar idea, but I believe that the habitat data were from habitat characterization of these birds in France. I would have to go back to that paper to double check that. But I think that in that case, we just use the exact same logic, which is basically just to summarize the logic of these papers, instead of defining habitats based on what we see, try to define the habitat based on what birds are in there. So we consider two habitats as being very different. The bird communities in these habitats are very different, and we measure the distance between habitats in terms of difference by these different communities. And so in that case, I think we use data from habitats in France. But indeed, you're right. The main reason was just to have this ability to run the analysis at a very broad scale. And also, obviously, it is very fast and very easy to access this data. That was part of it. We probably would not have done half of this work if we'd been working on getting more robust indices, but probably the data would be more relevant as well. It's definitely a work in progress, and it would be nice to further develop that. I agree.
Okay, thank you. Now, a very different question is that when we, and a kind of very recurrent with invasive species regulation, but when you have to kill tons of rats, do you have discussions about ethics with your students, and how do you lead these discussions? What are the kind of discussions you have? I mean, here explicitly, how do you speak with them, guide them between environmental ethics and animal ethics? And maybe it's not an approach that you really have in your mind or in your focus, because I perfectly understand that in the ecosystems you are working with, then there is really no doubt where you want to go. You want just to get rid of these rats, and that's what is needed to preserve the remaining species. So you go for it. But still, we are there facing, we want to preserve ecosystems by doing things to individual animals that are quite hard for them. So
No, it's an
how do you
excellent
position yourself, and how do you guide your students through that?
An excellent question that makes me realize that I probably am not guiding them enough through that. We haven't had that conversation so much, especially around the rat eradication, for a simple reason, which is we're not running this rat eradication, right? It's being run by an organization there. So it's happening whether we are participating or not. But it's clearly a key question, especially here in French Polynesia, where we don't have an ethics committee to respond to for the work we're conducting. So we're getting permits from the Direction de l'Environnement to work especially on species that are protected. But other than that, we never have to really think about that so much, which is clearly an issue. Then that's something we need to think about more. That's the way we have addressed it so far is not so much in that context, but more when we're walking around these seabird colonies, you always find birds that are either dead or dying, some that are clearly suffering, some that you feel like you could potentially do something, and that's always raising ethical questions. What do you do in that case? And in that case, we always are, there's been multiple times where the first time we're bringing a student in the field, there's a brown noddy covered in Pisonia seeds that's clearly not going to make it, but you could remove these Pisonia seeds and he would eventually have a chance, or maybe not, I'm not quite sure. But that's raising questions on whether we should intervene or not with this. Other ethical questions we are addressing are, I'm going to talk more about how we address that with the students after that, but it's just reminds me of another aspect, which is related to the way we're running these restoration programs. Today, now that the rats are gone from many of the islets in Tetiaroa, really we are hoping that the seabirds will come back, but the crabs are coming back much faster. So it really raises question on whether the seabirds are going to be able to settle down there where there's so many predatory crabs that are already there. And eventually there's a big colony that settles down, is going to diffuse the impact of the predatory crabs, but you need some to settle initially and be successful to make it work. So that's raising question on how much we need to intervene. Do we need to control the crabs there now to favor the arrival of these birds? Does it make any sense, or should we just let it work by itself? So these are questions that are more ethical, to my opinion, even though we can have a more science-based opinion on that, or science-informed decision. But yeah, I feel like each time we've had these experiences in the field, but we should probably have these interactions more often with the students, including in the lab, to prepare them for these experiences. We've just adapted to the reaction of the student. If we can tell that the student is just like, well, we can do something, but it's really just that one individual, and I don't care about the individual. What I care about is the general ecosystem. Then we go for it because that's our own personal philosophy. But if we can tell that the student really cares about that one bird being saved, and at least an attempt to save it, we'll just go for it. So that's the kind of plasticity we have adapting to the way the students are feeling about it. But we don't necessarily have this conversation about the ethic of it globally. So also because I believe some students have a very strong opinion about that, and I want to respect that opinion, even though I don't necessarily find that it is scientifically sound in these contexts. So maybe I've been avoiding that conversation sometimes, and it might be a bad idea. It's a good point.
Okay, thank you. So I feel I've nearly finished my time. I just to one, it's provocative, don't answer, and if we have time, you answer then. But it's in continental ecosystems, invasive species are not that an issue, less at least. We are more impacted by habitat change or climate change than in the islands. And there is a way of thinking, there are a group of scientists thinking about urban ecology and neo-ecosystems where indeed it's so disturbed that then why not making synthetic ecosystem made of super robust invasive species, and that's a way to rapidly rebuild ecosystems from scratch from post-agricultural lands. I guess for you it's no way to think this way, but if we have time, then I would be happy. And just the last one is, what is the little trick? Do you have a little trick, a little something that you tend to systematically do when you receive a new colleague or a new student and say, they will do that together? Like, do you bring them to the field? Do you bring them to a lecture? Is there something like a ritual you like to do when you receive a new staff?
A ritual, I'm not sure. So far, we've been lucky enough to bring the students to the field systematically. We are having our first student this year, that's Leo, who's going to arrive in a couple of weeks, who is the first one that's going to have a project that's entirely computer-based, but we're still hoping to bring him in the field at the end of his project. But so far, basically that would be it. I think I find it hard to bring students to work on existing data and not participate in the data collection, especially when we're here in French Polynesia. So that might be some kind of ritual in a way, I suspect. And just to respond very briefly to your other point, indeed, that these are conversations we're having a lot about the purposes of the restoration programs we're running. And there's some strong debates about how, not so much, there's no debate about whether we should just put a bunch of rats and invasive ants, etc., on the same island. There, we don't need to take care of it. There are debates about what species we should preserve and what is the purpose of these restoration programs. Are we running this restoration program to build a functional ecosystem, or is the purpose to preserve and build some backup populations of birds that are endangered in other areas? And we've had these really interesting conversations, sometimes very conflictual, including between scientists, about what species we should bring to Tetiaroa specifically, and whether we should integrate the local culture into that, which I believe should probably be where the decision should be taken before we can then inform them on which ones make sense, scientifically speaking, which one are going to survive it, for which one the habitat is available. But other people think differently and believe where we should pick the species that are at the highest risk of extinction to really have a backup population somewhere. So these debates are really important and interesting, I believe, but we really need to have, from my opinion, with the local population.
Thank you very much.
Thank you.
Okay, Lynn, it's your turn.
Okay, hi, lucky last. I've been listening to the conversation intensely, and I also read your thing, and I also had the same experience as Pierre-Yves in that I read it and I thought, oh my God, I'm never going to get through this. But I guess I thought, well, it's a whole life's work, and so you know, I need to pay attention and try and understand it. And I think in the end, you tied it all together quite nicely and made a really nice structure out of the whole thing, and you know, made it sound like you had a coherent life, which is a trick that I can relate to. And so well done to that. And yeah, it was really, really lovely. Open-ended questions. I'm going to ask you cane toad research, because that's the stuff that I'm most familiar with. But in the cane toad research, you keep saying that the tadpoles in Australia are more dense than the tadpoles in French Guiana. And I was just wondering if you actually measured that, or did you just base it on saying that there's, they're an invasive species, and so there's going to be millions of them at first? Like, how did you decide that there were more tadpoles in Australia?
So that's an excellent question. That's really what we believe is a driver, but we have some indication from old papers, from our own observations in the native range. There's some data in Australia as well on the number of tadpoles per pond. From our experience, especially in French Guiana, where we've been spending a few months looking for tadpoles around, we clearly can tell that there's way fewer there. But that's not data that we've published yet, actually, right? Yep. But yeah, that's I guess that's what I can tell you.
Yeah, because I can tell you that cane toad tadpoles in Australia vary from 1 or 2 in a giant water body to, to a whole pond, a whole dam full of tadpoles, so that you cannot see water. And so I just feel like the variation is so intense. Also, the tadpoles that come from Guyana are probably not very closely related to the toads in Australia phylogenetically, because the toads in Australia come from a different bit of South America, and there's probably adaptation across South America, I would think.
Yeah, that's an interesting point from the best of my knowledge, but probably wrong since you were saying it. I understood that they originally came from French Guiana and the Guyana, a mix of these two populations. But I'm probably wrong. I know that they went through a bunch of bottlenecks through the Caribbean and through Hawaii before eventually making it to Australia. So, but
I thought they came from Venezuela. But maybe we're both wrong, or maybe one of us is wrong. I don't know. I honestly thought they came from Venezuela, but I had a student who worked on toads in Florida, and they're totally different. They're much smaller. Their call is like 1 octave higher, and we just decided that because of the huge variation in Australia. I was wondering about the select pressure. I believe your results. I believe that they're more cannibalistic in Australia than they are in French Guiana. But I just wondered about the mechanism that you suggested was driving it.
Actually,
anyway.
one could have given to that is that we have measures of their genetics as well in the native range in French Guiana, and we did notice a lot of variation indeed. We did not identify, again, we should talk about that with Leanne Rollins rather than with me, but we did not identify clearly the genotype we see in Australia in the French Guianese toads that we sampled, but we find so much variation in very, very little areas that it suggests that this variation is there. And yeah.
Yeah, so so you mean that there was variation in the Australian toads or variation in the French Guianan toad?
Range, sorry. So it's, yeah, in the native range.
In the native range, yeah, okay. Even though the behavior was canalized, is that right?
No, that would be in Australia that the behavior is
In
slowly.
Australia, it was canalized. Okay. All right. So I guess in view of that, I wondered, you have done a lot of work, and you're still pretty young, and I imagine you'll do a lot more work. But I wondered, have you ever changed your mind about a result after you published it? Have you ever published a result and then thought to yourself, you know what? I don't think we were right about that. Has that happened to you yet?
Yeah, that's an interesting question. I don't think that it's happened to me. It's happened to me to feel like maybe I over-interpreted some results. Especially work on one paper on toads, actually, on the phénotypique plasticity of the developmental plasticity of tadpoles collected in different areas of Australia, where we measured their development rates according to the density of tadpoles, and looked at variation within the clutches in these developments plasticity as well. And I believe that in that case, our sample size was pretty limited. The conditions in which we tested them were not necessarily the most optimal conditions. So the results are what they are. The interpretation could probably have been more temperate, I believe, on this study. yeah, I think that might be the best way to respond to that question.
Okay, yeah, that's a good answer. And okay, so I wanted to ask some questions, same as everybody else, about supervision. And I was wondering what problems you've encountered with students, and like, can you put them in categories yet? And if so, you know, how do you deal with those problems?
So yeah, of course, we've encountered issues with students. I don't know that I can put them in categories yet. I'm potentially too young for that. We've mostly had students with which it was hard to get them to be independent enough to do the work without our intervention constantly. So with these students, the way we work is just by being there constantly. That was the type of student with which we've been just texting every day, giving phone calls continuously to make sure everything is fine, to make sure that they're still doing their jobs and that the project is making progress and they're not just going depressed alone in the field or anything like that. Which they're not alone in the field, shouldn't they?
Yes, I was just thinking that you're not allowed to go alone in the field. Any other problems, or just some people need huge amounts of support, other people much less?
I think so far that's the main issues we've had, and other issue we've had students that were definitely not interested and not motivated, in which case it's been a much more tricky issue for me, but I've been very lucky that it's happened very few times so far. So in that case, it's not much, from my perspective, there's not much to do. They just don't want to do it. Well, then just finish without doing it. That's a bit of a waste for both you and I, but that's, or try to push the student till the end to really try to figure out something that would enlighten some motivation, some interest in the work we're doing. But if really they're here to go surfing, can happen here in Tahiti, there's not much you can do about it. Eventually that happens, but we've been really lucky so far.
Yeah, okay, that's good. I just wondered, you've had a range of co-workers and supervisors over the years, and you actually mentioned one of them whose supervision you were interested in, and you followed their philosophy, or you used some of their philosophy. I was wondering, what did you take from your other supervisors, or are there other things you've taken from people you've worked with that you will apply in your own supervision?
Yeah. Yeah, it's a really interesting question. It is super viable. Each supervisor brought me something different here. I think with the supervisor you're referring to, there would be Louis Lefebvre and Daniel Stoll. I've had lots of interaction about how to supervise students. I've been seeing the way they were supervising them as well, and really appreciated their philosophy there. From other supervisors, I got different aspects. Like working with Rick, I've realized how well I'll never reach the productivity that Rick has, but I also don't really want that. That's not the way I want to do science, but I really appreciate the work he's able to put in writing papers so quickly, and how quickly he can imagine projects. And we can agree with him or not with the way he's doing it, but it's still remarkable in many ways. So that brought to me that perspective on the way of working that this lab has. So that's not the philosophy I want to follow, but that's a philosophy I'm really happy to have experienced to understand how it can work, to be in one of these labs where the lab leader is publishing 80 papers a year. But the other researchers, I've been very frustrated actually with my experience in Cambridge, for one easy reason. We were just there for a few months during college, so barely interacted with Daniel Field, even though he was a really young researcher. We're about the same age, and he had already a very dynamic research group around him. That's a paleontology lab as well. They all are very big geek paleontology birder guys, so really fundamentally passionate about birds, but in a very different area from what I was used to. They're really nice and interesting people to work with, and I was looking forward to seeing and sharing the experience he had of developing his own lab, interacting with his students, etc. But unfortunately, it was a bit tricky because it was COVID, and also because well, I was really happy to have that position with the IRD here in French Polynesia, but it also meant that I did not have any flexibility. I had to start right away here. So if I had to change something, I would probably change that to make that experience longer and benefit from the stronger input from that lab.
Okay, it's interesting that we share using Rick as an example of what you don't want to do. One thing I want to say, just also in response to Pierre-Yves, is the ethics thing. So killing things is not unethical. I've been the head of the ethics committee at JCU for 20 years. And so it's fine to kill things. I think that you made a great answer to that question, because the real concern is, what do you do about animals that are suffering? And, you know, do you intervene? And all those sorts of questions are the ethical questions, which is good. And then finally, welcome to ecoacoustics and having students sitting in the lab analyzing data, because you have so much data, you don't want them to go in the field. And so I wondered about ecoacoustics. You talked about bioacoustics a lot, and you talked about training recognizers for your individual species, which I was just wondering, you know, you will have data that allows you to look at community composition and lots more things than just bioacoustics and interactions between individuals. Do you have any plans to use that for anything?
Yeah, well,
we don't have a very specific plan yet. We've discussed a lot about looking at the space,
the acoustic space.
that the invasive are using as compared to the native birds or other organisms in there. So that's the type of question we thought we could address using more ecoacoustic approaches. So that's one aspect we've talked about, and that we talk about still these days with Antoine, with a new PhD student that's potentially going to address these questions. But we don't have a very strong plan either. If you can find any interest in these data, we're really happy to share that, definitely.
Yeah, we just need more people sitting in front of computers and analyzing all the data. Okay, thank you very much. Those are the questions that I had. I think the students are probably in good hands in the future.
Thank you.
Thanks, Lynn. So we're gonna go to a few questions in the room if people have them. Don't worry, not much longer, it's okay. We've got, I think, Jean-Claude first.
Okay, thank you. For scientists who don't have an HDR in their country, this kind of diploma degree may seem weird, but it is. And for French scientists like you, I know that it's often considered as a big waste of time, and it is. But unfortunately, it's not only this. It's also a good way to oblige the researcher to take a step back and to draw an overview of the first part of discovery. And on this basis, also to define clearly the main direction you want to take in the short and medium term. And both your written report and your one presentation today are very clear on these two aspects. And it shows that the exercise was a complete success. And congratulations for this. And along this line, I would like to know if the in-depth reflection you conducted for preparing this report has changed the way you perceive your own work, and does it help you to better position yourself in the local and international research system?
So I'm going to have two answers to that question. The first is no. I don't think that it really helps me making any progress with the way I'm seeing my research. I will just be perfectly honest with that. I don't think that this exercise was super useful to me, but it's probably because of my profile. I've been applying for jobs during many years before writing just 4 years ago. So like I've been doing that constantly in the past. It has been, though, very useful to make contact with the members of my committee, including especially people in bioacoustics and Pierre, of course, and that will, well, you'll realize, I think, that everybody here in that committee is working in bioacoustics or almost, and that's because we're really hoping to develop new collaboration in that area in the future. So that was a great opportunity, I think, to jump into that, and potentially we would have waited more time and be less bold with making contact with that regard here. So I think that's one key reason I found that exercise useful, but I still feel like it would have probably be more useful for me to just gather a committee so that we can write a project together or build something together, rather than having an evaluation committee, yet another one that I don't necessarily find so useful overall under the French system. I'm really happy to have this experience and to share my work, etc. I'm really grateful for all the time that's been put in that effort by all the committee members, but I still feel like the usefulness of it is questionable.
Okay. So totally otherwise, you have often worked on projects based on the participatory research approach. And I was wondering to which extent this kind of approach can be an effective means for collecting valuable data for your works, in particular for using them for calculating indices. I know that you have worked on behavioral plasticity index. I saw that to try to standardize the number of new behavior by the research report. I understand that. But how could we integrate other biases, such as the difference in the way information is collected from one study to another, for instance? This is already very difficult for research work, which are supposed to be implemented in a very rigorous and reproducible frame. And it seems yet more difficult using data from so I would like to
yeah, I'm not sure that I will fully respond to your question. This data set about innovation is actually not really participatory science. It's more of a meta-analysis, extracting information from published literature.
Yeah, that's a more general question. I took the example of this. I know that it was made differently.
It is a difficult question. I've used actual like citizen science a couple of times in my career. Maybe Pierre, sorry, would be the perfect person to respond to this question. I think his lab is doing a lot of that. But I've used that twice. Once during my PhD, where I was working on butterflies and aiming at understanding how different habitats were shaping the traits relating to dispersal in butterflies. And that worked very well because there was already a network of naturalists that was connected via the museum. So it was easy to reach to people to collect these butterflies and then to send them. We've tried something else here last year with Emma about the common minor, which worked much less well. It was really challenging. We tried to get people to send us information about these groups of common minor, where they are on the different islands, what kind of tree they're using, etc. I think in the end, we got information on 30 routes, including 27 that we detected and 3 that came from the citizen science. Usually it's going all the way right with this project. I believe the reason is that I don't know the local way of communicating yet. I'm not integrated well enough in the local communities. And Emma was also a student coming from the outside, so potentially we didn't know the way to contact. We tried to work with NGOs, but it didn't work so well. There's a lot of improvement there. In terms of actual data collection, in that case, we can go and trust the data in the field. So I think it's going to be really useful, that framework. And I may again not respond to your question. I think it may not, in some cases, with this kind of project, we imagine also running some project with birds, especially minor, with different color bands, and then get school, get students, get people in general, citizens, to send us information on which birds they've seen where, to attract their attention to these species. And it may not bring very relevant scientific data, but I think it may bring a lot in terms of raising awareness of these questions. And that may be where the objective is, more than in the science. That's, to my opinion, equally important. Thank you.
Yeah. Okay.
Thank you, Simon, for proposing to participate to your jury. It offers me the opportunity to discover the impressive work you've done last 2 decades. I'm really happy to be here this morning with you. Like me then now, but congratulations first for what you've done. So you have already answered a lot of questions. So first, I only want to compliment what you've done, but I want just to add something on the question on ethics you asked people previously. I would only make the jury know that you were talking about rat eradication, but you did not mention ant eradication, and saying that you were really we had discussion about the impact of the treatments and the polymer used on in ant eradication. Sorry, and you were really we tried to collaborate because you were concerned by adding a lot of polymer in the environment. I'm working on plastic pollution, so it's the reason why we try to discuss more. So you are, I would say that you are really the ethics, you consider ethics as something very important in your work, but you did not mention it because I wanted only to make this clear for all. Here, I only wanted to ask you one question. According to you, or how habitat restoration can maybe influence behavioral plasticity? Do you have any response answer on that?
Yeah, that's a it's a
Maybe a
broad
big
question,
question,
yeah.
but only just the first element.
I guess I can just tell you about one example that we're working on in Tetiaroa, where we're measuring how the crabs are responding to the rat and ant removal. And there's one crab species, a ghost crab, we've been working a lot on, and where we noticed a change in behavior immediately after the rat eradication. So we can tell that in that case, it's some level of plastic response. So they altered the behavior. Once the rats were gone, we could tell that they changed the moment they're active. So they didn't have the same activity phenology than the 24-hour cycle. They used, when the rats were present, they were avoiding the activity peak of activity of the rats to not be eaten, basically. And a few weeks after the rats were gone, we could tell that they could respond to that by a change in their behavior. So that was an interesting, I believe, way to show that it's not just the population that's changing, it's a behavior of the individuals as well. That has, in that case, I'm really happy actually that you're asking that question. In that case, it also has severe implications for the way we're monitoring the response of these populations. So the example of these crabs, the way people are monitoring crabs on atolls, usually you just run transect. So you count the number of active crabs at a given time of the day when you know that they're supposed to be active. And you just do it at the same time everywhere, before and after the eradication eventually, if you want to see if they react to that. But if the crabs change their period of activity or change how often they're active because of the rats, then you're not assessing a change in population size, you're assessing both the change in population size and activity, so behavior as well. So that's, I think, some information that's important to have to probably improve our monitoring protocols in that case.
I also asked us about how the time, the scale, I would like, meaning that if are you solving a problem when you restore an habitat, or you add more perturbation, and the response you look at can be short-term or long-term response, and maybe this is like a cycle. When you restore something, you modify the behavioral plasticity, or it's only adaptative. So adaptation might be more short for short period than plasticity. Or what is the scale? What can be the scale of when you to discriminate behavioral plasticity than adaptation?
It's probably something we can count in according to the generation time of these species. Obviously, so for crab, it's probably going to go faster. Plasticity would probably come first, and then a fixed response may emerge, more adaptive response, a genetic change might emerge later on. That's what I would expect. And that's what most of the theory is expecting as well, and results are suggesting that this is the order in which it would occur. Yeah. So here, potentially with these crabs, we can imagine that the ones that were able to plastically respond to the removal of rats were finding more food and were more likely to find mates. The ones that were not plastic and kept staying in their burrow for a really long period of time probably will be eliminated eventually from these populations. So right now we have that plastic response that will be favored and selected. Maybe one day, because it hasn't been rats generation to generation, it will stop being plastic and they just by default be active at these moments.
The cool thing about going very last is that everybody's asked all the questions, and I have nothing left to ask now. I mean, I have lots of questions, but like we've been talking for a few days, and it's been so awesome. Just like you said, maybe, you know, silver lining to the cloud of the HDR, I think anyways, that I got to come here and have discussions with you and Jana and your students as well. And they all seem happy, so that's great, that's a good start, right? What do you suggest, how do you suggest the ability of supervisors should be assessed as an alternative to this process?
Good question. I don't know if I have the perfect answer, but I do have an answer to that. I think one thing that's been done a lot in North America, especially, is these exit polls. You just ask your students what they think at the end of it, and it's not you personally who's asking it; it's the university. So there's a way for it to be a bit more objective. They don't have to respond to you that they were really happy with the supervision just to be nice to you because you may or may not write a letter for them. So I think some neutral exit polls to know what the students felt about you would be important. Not only that, though, because of course sometimes it doesn't work well with a student and it's not going to be very objective in any case. So I think, but that would be an aspect. The opinion of your colleagues on the way you're supervising students, they're the ones who see you interacting with the students as well around, would be also probably rewarding. Of course, I think you need to some extent to look at the quality of the research. But I feel like this exercise right now is almost entirely focused on that. I was really happy that the committee members asked a lot of questions about the students, but that's not so much part of what we are evaluating on, which is a surprising process to me, given that the conclusion of that will be whether or not I'm allowed to supervise students. So I think that's probably what I would look for the opinions on the way I've been supervising students in the past from both the students and the people who see me supervising. It's really difficult to be neutral and objective in that regard. I get it.
Yeah.
And it probably would be more work for people other than me to gather this information. So I understand that it's not so simple. I feel like there might be some space to do that.
Yeah, I like super impressed with, it doesn't seem that you have sacrificed quality for quantity. It's just both in your research, which I like. Yeah, it's awesome. My other question is, if you didn't work on birds, what would be the next, or cane toads, I guess, what would be the next taxa, do you think, that you would like, just like, you're like, oh, you know what, that would be really cool to look at, especially in terms of, say, cognition, plasticity, and that sort of thing. Maybe it's underrepresented, or maybe you just have found in your experiences, like you're looking over and while you're doing your bird work going, oh, that's so cool. Is there any other taxa that you think like kind of piques your interest in terms of your research?
Yeah, I think right now, terrestrial crabs, just fascinating. There's so much to do with these guys. They're poorly known. Each time we're reading new papers about them, discovering new things in the field as well. Not so long ago, we realized that just talking about that, these ghost crabs are also acoustically active. We can hear them singing from their burrows, which is pretty amazing as well, very interesting. These are poorly known behaviors as well, so I think there's a lot to discover there.
Yeah, awesome.
But let's see with birds for now as well. I want leaves of birds.
That's fair. Great, thank you so much. I think that's it then for the questions. Thank you, everybody, for listening for this whole time. You guys are all still awake. Excellent. And thank you also to the committee. We kick them out now again. So, do you like serve in? Okay, just to reward you for your attendance. So.